In this lecture, we'll discuss an introduction to the cell. Every human being is made of cells. In fact, every living organism on the planet is composed of cells. And these cells are all able to do certain basic processes. We can do cellular metabolism, which involves both anabolic and catabolic reactions. Anabolic reactions are used to build molecules, while catabolic reactions are used to break down molecules. And cell metabolism is just a summation of all of those chemical reactions combined. Cells are capable of transport. This includes both moving substances into and out of the cell, as well as moving things within the cell itself. So just for an example, moving a substance from the left side of the cell to the right side of the cell. Cells are capable of communication through chemical and electrical signals. Cells are capable of talking with other cells that are close by to them, as well as cells that could be farther away from them as well. And cells are capable of reproduction. They can do this either in a sexual or asexual reproduction manner. So if we're talking about cell replication specifically, we have to include DNA replication. Any new cell that will be created must contain brand new DNA. There are four main steps to DNA replication. First would be to unwind the double helix itself. We need an enzyme called helicase to help us do that. When we start to unwind the double helix, we don't want it to go right back together. So step two is we have proteins. They are called single-strand binding proteins, and they bind to each part of the helix and to help keep the helix separated. This allows for another enzyme, DNA polymerase, to come in on each individual helix strand and start creating new DNA. DNA replication only goes in one direction, from a 5' end to a 3' end. So the 5' to 3', if you want to think about it as maybe like a left to a right, like what we read. We read from left to right. Okay. So we always go 5' to 3'. Now this gives us the problem. Okay. We have our two separate strands of DNA that we have separated. One of them, it's very easy to create our new DNA from that 5' to 3'. It's no big deal. We call this the leading strand. The other strand, however, we're going to have to work backwards in order to achieve the 5' to 3' direction. We call this the lagging strand. It is made in a discontinuous fashion. We have to wait and open up the double helix before we can get to enough bases to create a new section on this lagging strand. And anytime we are making new DNA, we actually start with a very small RNA primer. So we have yet another enzyme, RNA polymerase, creates the small RNA primer. It really just kind of gets it going. And then DNA polymerase comes behind and starts adding on new DNA nucleotides. The DNA polymerase will eventually go back and remove those RNA primers and replace them with DNA nucleotides as well. Now, we mentioned on the lagging strand, the DNA is created discontinuously, so we end up with these fragments or these chunks, and these are called Okazaki fragments. We have yet another enzyme that takes those individual fragments and bonds them together to create one continuous strand on the lagging strand as well. So that was a lot of words. Let's look at a nice picture. So we have our DNA double helix. We see our 5' ends and our 3' end. So our top strand runs 5 to 3. And our bottom strand would run 3 to 5. Okay. These numbers, these 5s and these 3s, it's the number of carbons. We don't want to get too bogged down into a lot of detail. But if, like I said, if you want to think about the 5 being like the left side of a page and we read from left to right, so the 3 would be the right side of the page. Remember, we're going to go from 5 to 3 always. Okay. This bottom strand, it's kind of like the page being upside down, and so the 5 is now on the opposite side that it normally would be. So when we go to make DNA, we can only go from 5 to 3. So we start with that top strand, that leading strand. So notice that we've had our helicase come in and open everything up, and our single strand binding proteins are keeping everything separate. So our DNA polymerase can come in here and start laying down DNA nucleotides. We would have started way back here, and he would just chug along, adding nucleotide after nucleotide, creating that 5 to that 3. Okay. We said our bottom strand would be a little bit different. Okay. If we're going to create that 5 to 3, we would have to start over here. Okay. Well, if we look at this, which was originally created, we're like, okay, 5 to 3, 5 to 3, but notice we've done this in chunks. Okay, we mentioned that. Why are we having to do it in chunks? Because we need to get down here to make our next section. Well, helicase hasn't had an opportunity to come in here and open that section for us yet. So every time helicase opens the new section, then DNA polymerase can come in and make more of that section. Okay, I can replicate that section. We did mention that we start with RNA primers. We have the RNA polymerase. We said that DNA polymerase will eventually remove that RNA primer. So notice here at the beginning we have no more primer. And we mentioned that DNA ligase would bond these two fragments together. Okay, so we would end up, eventually, we would end up with one nice continuous strand on the bottom as well as on the top. Okay. Now, we need to be able to come up with a new sequence of DNA from an old sequence. So if we take this sequence, okay, here's our example. So you've got your notes, or just jot this down on a piece of paper real quick. Okay. We're going to take this one strand and we are going to make the complementary strand. Okay. We have base pairing rules. So adenines pair with thymines, cytosines pair with guanines. A's and T's, G's and C's. Okay. So anytime you see an A on your original strand, your complementary strand would have a T. Okay, so on and so forth. Pause your video and do this example. Make sure you understand A's and T's, G's and C's. When you're ready to check your work, restart your video, check your work, and then let's move on. All right, so let's check our work. Okay, as you can see, A's and T's, G's and C's. So ATT becomes TAA, CTC becomes GAG, right on down the line. Okay. You do need to remember A's and T's, G's and C's for your exam, your quiz, all that good stuff. Okay. Now, not only do we need to make new DNA, but we're also going to need to make some RNA every once in a while. So let's talk about protein synthesis. Now, RNA synthesis, or excuse me, protein synthesis. The DNA that we have inside of our cells, what is it good for? Everybody knows, oh, DNA makes us who we are. Well, what do we do with that DNA? It's really about turning that DNA into proteins. Okay, so protein synthesis. We take a DNA strand and we turn it into a set of instructions for making a protein. Those instructions, the process of making those instructions is called transcription. Okay, so the word transcribe kind of means to write. So we're going to take our DNA and we're going to copy it down into our RNA instructions. We're going to transcribe it. This is a 3-step process. And we start with initiation, we move to elongation, and we finish with termination. And if you know what the word initiate means, it means to start. The word elongation means to make longer, basically. And termination means to stop. So we're going to start, we're going to make our strand longer, and we're going to stop. So the steps, they're not that bad. So who is doing the starting? You have an enzyme, RNA polymerase. It attaches to a very particular region of the DNA. It's called a promoter. This is the beginning of our instructions. So our RNA polymerase binds to our promoter. Okay, it separates our double helix so we can really get in there, do what we need to do. Then we move to elongation. We start reading those DNA instructions and we start copying them down as RNA instructions. So RNA polymerase starts to add complementary RNA nucleotides to growing strands. Now, what's the difference between DNA nucleotides and RNA nucleotides? Well, those base pairs we just talked about. Remember, we said A's and T's, G's and C's. Okay. RNA is a little bit different. RNA does not have thymine. It has no T's. Instead, it has a uracil. It has a U. Okay. So if you see a strand with T's in it, automatically DNA. Boom. You know, you're done. You see a strand of letters that has uracil or U's in it, boom, RNA, you're done. You know what's up. Okay. So RNA polymerase, it continues reading. It continues to transcribe our instructions into our RNA sequence. It keeps going until it hits a terminator sequence, which is kind of like a stop sign. Okay. This signals that we have finished copying down our instructions. We allow the double helix to reform, and we are left with one strand of RNA instructions. Then we have to take those instructions and make a protein. Okay. Now, before we make our protein, let's make sure we can take a strand of DNA and turn it into RNA. Notice that we have new rules. Remember, I mentioned that RNA has no thymine, it has uracil instead. And so anytime you see an A in a DNA strand, it would pair with a U or a uracil in that RNA strand. So again, pause your video, jot down your answers when you're ready. Let's come back and check our work and make sure we're off to a good start. Okay, let's check our work. Okay. So ATT becomes UAA. CTC becomes GAG, so forth and so on. Notice our DNA does have the thymines, while our RNA has uracil instead. Okay? So now that we have our RNA sequence, our set of protein instructions, let's turn that into a protein. This is known as translation. You will notice that the 3 steps of translation are named the same thing: initiation, elongation, termination. We're going to have the same overall functions. We're going to start, we're going to make something longer, and then we're going to eventually stop. But our players are a little bit different here. You are going to start with ribosomes. Okay. If you already know what ribosomes do, great. If not, let me mention ribosomes make proteins. It's literally their job. So ribosomes are going to get together with a tRNA molecule, and they are going to bind to our mRNA strand. Okay, so we have lots of different parts here. Okay, so mRNA is messenger RNA. That is the set of instructions that we just created. Okay, so we have our instructions, our mRNA. Okay, ribosomes are the organelles that make proteins. Okay, so our little protein factories are going to combine with our instructions. We're going to join together, and we need somebody to bring in the components, the amino acids. That's what tRNA does, transfer RNA. Okay, everybody is going to come together at what we call the start codon. Okay, now a codon is a set of 3 nucleotides, 3 bases. Okay. If we go back real quick, notice that we group these into 3s. So UAA is a codon, GAG is a codon. Okay. So we're going to have to find one of these in particular. One of these is going to be special. That is our start codon. So once we find our start codon, our ribosome comes together, our tRNA brings in our first amino acid. So remember, what are proteins made of? They're made of amino acids. So tRNA brings in that first amino acid, and then we keep going. Okay, we move on to our next codon. Our tRNA brings in our next amino acid. We bond them together. We do that over and over and over. We build a polypeptide chain. We continue to do that until everybody comes to a stop codon. Okay. Fairly self-explanatory. It is time to stop. We have reached the end of the instructions. So once we hit that stop codon, the ribosomes will disassemble. Okay. The polypeptide that we have formed, it will be released. It will move on to other parts of the cell where it can be modified until it is fully functional. And because that's a lot of words, let's look at our picture. Okay. So this pink section is our mRNA. Remember, our messenger RNA, our message, our set of instructions. AUG is our start codon. AUG is always the start codon. It's the only start codon. Okay. So we've got our ribosome parts, our little factory. And we've got our tRNA bringing in our first amino acid. So we're getting all our pieces together. That's what initiation is. Everybody's coming together. Okay. Once we have everybody coming together at our start codon, we move on to the next codon, which is elongation. And notice we just keep going down the line. One codon at a time means one amino acid at a time. We're forming those peptide bonds. We're growing our polypeptide chain until we hit that stop codon. Now there are 3 stop codons, UAG, UGA, and UAA. Okay. And these do not code for amino acids. They're literally just stop signs. Okay, so one is a dead end, one is a stop sign, one is a red light. Okay, they all look a little bit different, but it's the same result. When that happens, okay, that tells the ribosome that we're done. That tells the tRNA that we're not bringing in any more amino acids. And everything just kind of stops. So your polypeptide has been finished. Your ribosome can be recycled. Your tRNAs can be recycled. We might even use that piece of mRNA again. Maybe we need to make more than one of these polypeptides. And we can do this as many times as we need to. Now, how do we know which amino acids to bring in when? It's all about these codons. Okay, each codon represents a different amino acid. Now, we've already mentioned that there are only 20 amino acids. There are quite a few extra codons. So several codons will code for the same amino acid. For example, if we look at this box, glycine, which is one of the 20 amino acids. Each one of these 4 different codons, okay, all 4 of these codons, they all code for glycine. So there's a lot of overlap, and that's okay. Now, here's what you don't need to panic about. Do not memorize this chart, this genetic code table. Don't do it. Don't do it. Okay? You will have this on your quizzes. You will have this on your exam. I have not memorized this, nor do I plan on ever memorizing it if I can get away with that. Okay? You just have to know how to use it. So let's practice using it. We're going to do the first one together, and then you can pause your video, jot down the rest of your answers. We'll come back and check our work. Okay? So you will notice AUG. I did mention that that is our start codon, but let's make sure we could find it on our chart anyway. So we have 3 letters, and we have 3 sides. First side, second side, third side to our chart or our table. So we start with our first letter, A. We start on our first side. So we find our A. Okay. So we have taken this entire table and we have narrowed it down to something in this row. Okay. So we have immediately disqualified everybody else. We're just in this one row. Everything in this row, look, A, A, A. Everything in this row starts with an A. And then we move to our second letter, is a U. Okay. So we're staying in this row, but we have to go up top and find our U, which is here. So where the A and the U meet, which would be this box, we have narrowed it down to this box. Okay. And then we have our third letter, which is G. You could do this one of two ways. You could go all the way back to the third side, find the G, go all the way over. And if you've got your finger on it, you got to hope your finger doesn't move and you end up in the wrong box. Okay. Or, you know you're in this box. There's only 4 things in the box. Just go one at a time. Where is my G? Boop, there's my G. A-U-G. This codes for methionine. Okay. Again, our start codon is always AUG. Our first amino acid is always methionine. Okay. But at least that gives us an idea of how this chart works. Now, we do need to practice. So, pause your video. Take the rest of these codons. Use this chart. Figure out what order these amino acids need to go in. When you're ready, let's come back and check your work. Make sure we are professionals and experts on this.
There you go.
All right, here we go. Let's check our work. All right, so we did the first one together. We had methionine was AUG. Okay. Next was CCA, so we had C and another C. CCA is proline. In fact, everything in this box was proline. Okay. Then we have ACGA, CG. Again, we got lucky. Everything in this box was threonine. GAG would be GA. So we've got our GA, we come down, GAG specifically codes for glutamic acid. CGG, we've got our C, we got to find our G. Where do those intersect? That would be this box. So CGA, these all code for arginine. Now, CGG is arginine. CAA, let's see, CA. All right, so we got our CA box. CAA would be glutamine. Next is AUC. A and U, we're in here. So AUC would be isoleucine. And then UUC, U, we come down, UUC would be phenylalanine. Now, if we did UGA, UG, and we come down, UGA is a stop codon. Okay. Again, you can write stop codon if it makes you feel a little better. If you're like, well, this is here, I feel like I need something down here, that's okay. But leaving it out is also perfectly okay, because keep in mind, stop codon, that's not an amino acid. It's just a stop sign. Okay. All right, if you need more practice with this, please don't hesitate to reach out. And